Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. Engineers design a car to be just right from the production line. If you inspect the car after it has run 50,000 kilometers, the machine parts are exactly the same, except wear and tear. And when the wear and tear become too much, the car simply stops working. We tend to have the same mental image for a nerve cell. It is built during development and then stays the same unless something bad happens to it. This is sort of true, but what exactly stays the same? A so-called fast spiking interneuron in cortex is an example of a cell that is known to maintain a characteristic high-frequency non-adapting spiking pattern stably over long periods. So the firing characteristics of this neuron stays the same. But what about the building blocks? Like any neuron, the firing characteristics of a fast spiking interneuron is determined by the particular combination of ion channels in the membrane. But is the particular combination of ion channels kept the same? Not necessarily, since there are many combinations of ion channels that can give the neurons the same firing characteristics. In modeling terms, we say that the solution to give a neuron a particular firing characteristic is not unique. It is so-called degenerate. This degeneracy is helpful for the fast spiking interneuron to keep its functional identity. If, say, one of the ion channels no longer functions properly, then the neuron can find a new combination of ion channels to maintain their identity. But how does the neuron know what its identity is? According to the work and ideas of the present guest, Tim O'Leary, the identity may be encoded in the rules that determines how much of each ion channel is produced by the cell. That is, by the production rules of the proteins that make up the ion channels. Together with colleagues, Tim published a really nice paper on this topic back in 2014 that made an immediate impression on me when I read it. The standard approach when making biophysically detailed neuron models is to fit ion channel densities to make the neuron model fit electrophysiological data. Maybe an alternative could be to instead find or fit these ion channel production rules and then let the model itself compute the right ion channel densities. This approach also allows for the direct investigation of how homeostasis work at the neuron level. That is, how neurons can find back to their appropriate working point if something bad happens to some of the ion channels. In the podcast, we also talk about the different but conceptually related phenomenon that Tim has worked on and coined representational drift. In experiment, it has been found that neuronal representations of, for example, learned tasks reconfigure or drift over time. So while a mouse can be able to do a learned task like navigating a maze to get to food perfectly over weeks, the detailed neuronal activity involved in doing this task may change. For example, a neuron whose activity is always high when the mouse do the task the first week may be quiet when the mouse do the same task the third week. So what is fixed is the ability to do the task, not the detailed underlying circuit behind it. How and why does such representational drift come about? If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I really appreciate all of you who are already Patreon supporters. Thanks a lot. And I will be most grateful if more join in to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Tim. Yeah, so welcome to the Theoretical Neuroscience Podcast, Tim O'Leary.
I'm very happy to be here. Thanks for inviting me.
Very good. So first, a little bit about, you are now at Department of Engineering at Cambridge. Is that so?
That's correct.
Yeah. You've been there for a few years?
10 years now.
10 years.
It's gone quickly. Yeah.
Yeah, because I first came across your work in this really cool paper from 2014, where you sort of had this, I guess it was a paper in Alex Williams, Alessio Franchi, and in Eve Marder's group. You were all in Eve Marder's group at the time. And because it had this really, this really cool idea, I think I remember it really made an impact on me because it addressed this question of how does a neuron keep its identity? How does a bursting neuron, like LA5 bursting pyramidal cell, how does it know that it is a bursting neuron? So that if something bad happens to it, that it sort of remains a bursting neuron and maybe sort of comes back to the bursting ways after some kind of period, right? So I guess that was like a main topic of that paper, right?
Yeah, so that was certainly part of it was the question of cellular identity. And as you said, one of the obvious problems with any type of feedback regulation, especially if you think about something like a neuron, which has lots of different types of ion channels, there are many options for correcting. And an immediate question is, how does the neuron not get lost? How does it lose its identity at the same time as adapting to all of the different kinds of insults or everyday life perturbations that it will experience?
So because I guess a starting point was like previous work from Eve Marder's group, where like this degeneracy of neurons. There are so many ways that you can combine ion channels to give a particular like functional behavior of a neuron, right?
Yes, and I'd say that this observation is certainly not unique to neurons. It's not even unique to biology. It's something engineers have to deal with as well. There are often many different ways of assembling an equivalent circuit to perform a certain function. So how it actually ends up being assembled as a result of some design constraints and some decisions.
I guess I've heard this quote from Tolstoy that there's like so many ways for a family to be miserable.
Yeah. Yeah. You can homeostatically regulate misery in many different ways. That's right. And happiness, I would add.
Well, he claimed that it was only one way to be happy.
Oh, really? Okay.
So anyway, so that's why I think he focused on
misery is more degenerate than happiness. That's
that was, I think, his claim. Anyway, so because often when we do, I mean, I think the most typical way to model neurons, right, is maybe like biophysically detailed neuron modeling with ion channels, like Hodgkin-Huxley style, maybe with dendrites and so on, that you have to zoom some set points, like, for example, resting potentials of the cell, and maybe some ion concentrations at rest. But what was really cool about this paper is that you really went below there, and so where does that set point, where do these set points come from?
Yes, to some extent, yeah. So in the end, a set point is really just an equilibrium of some kind. So you need some kind of push-pull dynamic that settles down at an equilibrium. And the more things you connect to that in the right way, the more you can maintain that equilibrium. Sometimes at the cost of other things not being maintained, and that was one of the other themes of the paper, is that this can and does go wrong. So just more broadly in the field, there's decades of observations, some of which might fall under the heading confirmation bias, where you see various publications showing that things homeostatically adapt. When you speak to physiologists, as often as I do, occasionally you speak to someone who says, "Oh, I don't believe that that's true." in the system or the specific setup that we studied, things like this.
Oh,
We
so
didn't see
they don't believe in your
any.
models,
Well,
or
no, it's not so much that they question the dictum that every aspect of the nervous system is homeostatic, and of
ah,
course it can't be really.
mm,
It has
mm,
limits.
yeah,
Otherwise, we would never get sick. So there are these limits. They're interesting. I would say they're understudied because the literature so far, for obvious reasons, has focused on positive examples of
okay,
homeostasis.
thanks.
It's a bit harder to study where, why, and when, and how homeostasis
Ah,
fails.
ah. I guess there's this other principle. It's what's called allostasis, right? Where it's more about
Allostasis, yeah.
that's predicting,
Yeah,
adapting to the future a little more
yes,
than coming back to baseline. Yeah.
yeah. So allostasis is an interesting concept. My own view on it is it doesn't add that much. It adds another element, but
Mm,
nothing that you wouldn't have seen outside of standard control engineering. So you
okay.
often have predictive elements
Yeah. Mm.
if you want to
So
maintain some
I
kind of set
mean,
point as well.
you are in an engineering department, and you also have some kind of background in control engineering, or was that a huge
Well,
field?
I studied it. So my background is mathematics. So I studied what's called modern control theory.
Ah,
It's actually old
yeah.
control theory,
That's like
and
modern physics. That's also
yeah.
quite old.
But then I didn't really have much contact with it until I came back
Uh,
to essentially a physical sciences department, an engineering department, and now
why don't you?
I'm
Yeah, so why
based
don't you?
in the
Sorry.
control group.
Why don't
So
you tell a
I
little
have
bit?
colleagues who really study this for real, and they've taught me a lot actually.
Yeah. So why don't you tell a little bit about your background? Because it's yeah, in your I mean scientific background,
Sure.
your education, and
Yeah. So I started off in mathematics, and I started off a PhD in pure mathematics in hyperbolic geometry. Interestingly, I had the same PhD supervisor at the same time as Dan Goodman, who was another
oh,
theoretical neuroscientist.
oh, yeah, yeah,
And somehow during those years, so he actually finished his PhD. I left mine to pursue neuroscience. He
okay.
left maths after finishing the PhD
So
to pursue neuroscience. So
I
there was
see.
something in the air at that
Yeah,
time, I think.
yeah,
Um, Essentially, what I felt was that all of the interesting problems got mopped up in the 70s in
uh-huh,
the field that I was studying, and it
yeah.
didn't feel exciting and vibrant, at least in the
No.
field that I was
So
in.
where was this?
Where? In
Yeah.
University of Warwick,
Okay.
UK.
Yeah. Mm.
Um, so I switched to neuroscience. I got interested in um, the biophysics, biophysical aspect. That's essentially an easy entry point for someone who has um, a sort of maths and physics background. Um, and I actually decided to go fully into experimental uh, neurobiology. And my first contact with that was actually doing single channel recordings. So patch clamping individual ion channels, essentially, in this
Mm.
case, NMDA receptors.
Wow.
And that was really fascinating and an eye opener because
Mm.
you realize the um, pains you have to go to get interpretable measurements and all of the things that need to land in the right place for you to get data that you
Yeah.
can actually understand. And that's
Mm.
just to study a single ion channel. So
Mm.
then piecing together all the rest of it
Yeah.
is really an enormous task.
Mm.
But then from there, I spent a bit more time. I moved from single ion channels up to whole neurons and eventually
Mm.
networks. And then as I moved to a postdoc with Eve Marder, that was an opportunity to go back to the mathematical modeling, really, which I had
Mm.
done a little bit in my
Yeah.
PhD. But it's
Yeah.
very hard to do full on experimental neuroscience
Mm.
and theory. At least it is for me. I don't know how people manage that.
Yeah. No, I don't know either. I never tried to do experiments. So, but I think it's also. I mean, Eve Marder has been very good at sort of involving good modelers or theoreticians
Yeah.
like you, because
Well,
I actually yeah.
she basically brought Larry Abbott into the field.
Yeah.
who at
Yeah.
that point was he was in the physics department in Brandeis,
Yeah.
and
Yeah.
Eve's genius is actually telling people what the relevant question is and
Mm.
finding the
Yeah.
skills that they have that can answer that
Mm.
question.
Yeah. Yeah.
And
Ah.
she's not concerned about someone's background or someone's field
No.
if they can say something useful
Yeah.
and helpful and
Yeah.
communicate it well.
I think
That's the
that's a
important
very
thing.
important quality if in sort of finding efficient ways to have people with different trainings collaborate to ask like to answer particular questions. And
Yes.
that's it's not a trivial thing. It's just
No.
about not just about inviting people with different backgrounds in. It's sort of
No,
making them
that usually
communicate.
doesn't work.
Yeah. No.
There's actually a brilliant essay I love by someone called Sean Eddy, who wrote
Mm.
this essay. I think it's in PLOS Biology, called Antidisciplinary Science.
Okay.
And
Yeah.
his point is that actually, if you've got an interdisciplinary problem, and most
Mm.
things in biology, certainly neurobiology, are interdisciplinary, if you think that the way to solve that is just by getting enough people from different fields and just sticking them in a room to work together, it will fail, because they'll bring
Mm.
their culture, their own questions, and their own
Yeah.
priorities to the
Yeah.
question.
Yeah.
Instead, what you have to do is find people who are actually genuinely interdisciplinary themselves, who are
Mm.
actually searching for questions outside their field.
Yeah. Uh, yeah. Yeah, that's I'll look that up. I can make a link at the homepage of the episode so that people can look it up also. So cool. So back to sort of the 2014 paper, because there I think most people who sort of when you do biophysical modeling Hodgkin-Huxley style, then you have to sort of to assume some numbers for the densities of these ion channel conductances, or essentially density of ion channels. And then you sort of, well, it's like a combination of maybe getting input from experiments or just using them as fitting variables. But you, and this is of course quite unsatisfactory, because at least I think, because we know that these things change over time, right? And it's like what, yeah, and it's that
Yeah.
you cannot really, these degenerate systems, you cannot really fit parameters by using different experiments at a time, right? You have to sort of to actually get all the parameters from the same experiment, if anything. And but you sort of went, which I think was really cool, is that you went one level below and looked at, actually made a model for how these different ion channels proteins are made, right? So then,
Yeah.
and then you need some kind of link between what the neuron is doing
Mm-hmm.
and the transcription or the making of ion channels, right?
Yeah.
And that's where this intracellular calcium comes in. Why? So
That's
why
right.
that? Right? So why intracellular calcium?
Right. So there's lots of ways of answering this. The short one is that even if you look outside neurons, you look at other cell types, calcium is this key player in lots of intracellular signaling. And its role in regulating ion channel expression is preserved, again, outside neurons, even outside
Mm.
excitable tissue. Calcium is
Mhm.
involved in major transcriptional pathways. For example, it
Mm.
activates genes and so
Mm.
on. So there's abundant evidence. And on top of that, actually, my PhD, I found some evidence of this myself. I actually blocked a major calcium pathway and found that hippocampal neurons lose their ability to
Ah.
homeostatically adjust intrinsic
Ah.
conductances.
Ah.
So wherever you look, calcium is sort of implicated in this process. That said, the model that we developed is really a cartoon.
Sure.
It's pretty feeble on the level of detail, but
Mm.
our attempt was to really capture the essence of what's going on, and just to say, well, there is this bulk signal, and many things respond to that signal. So essentially, there's kind of a bottleneck where you've got lots of different processes affecting this one signal. And similarly, then the signal affects lots of actuators, or lots of genes, which then affect neurons' dynamics. And this pinch point is actually what was, for me, the interesting aspect of all of this. That when you have that pinch point, you necessarily have a high degree of degeneracy. The rest of the model then simply assumes that there is a constraint on the rate at which these different channel types are expressed. And that's all you need, actually. If you just regulate things by this one variable, but assume
which is the intracellular calcium concentration.
Intracellular calcium, yeah. But you assume that the relative expression rates of these different channels is preserved. Then you get for free a lot of interesting things. One, you get correlated expression in the channels, which is something that's observed, at least where it's possible to measure it. And as a consequence of that, you get preserved neural dynamics, if you like. So, as you mentioned, things like a bursting neuron will remain a bursting neuron, and so on. Within limits. All of this works within limits. Outside of certain limits, if you massively perturb the system, of course, it won't recover in the way that you expect.
Because one thing is sort of, I mean, you have this in this, described in the model in the paper, of course, that you have this quite simple dynamics of how sort of like this messenger RNA, mRNA, are transcribed, right? Or transcribed. That's called, or I'm getting transcribed from the DNA, and then how the mRNA is then translated into protein.
Yes.
So that's like simple first order, like chemical.
Those are just first order processes. This is like genetic modeling 101. It's the simplest possible model you could write down. That's not offensive.
No, so that's true. So this is like, and then you have this key thing that in the source term for the DNA transcription or this mRNA transcription, you have sort of like this calcium concentration. It's like this intracellular calcium concentration, and so that is rates. And because one thing which is maybe surprising is that you have one sort of master regulator, the intracellular calcium concentration, and from that you get all kinds of different you get different densities of different ion channels. So you have sometimes one regulator that regulates all these different levels of ion channel sort of production. So where does this sort of like, how can you regulate so many things with what is different between the ion channels then?
Simply the rate at which they're regulated. So preserving that rate, as long as all of the action happens within some volume in conductance space, then as long as you preserve those relative rates, you end up with preserved steady state ratios of conductances.
So it's actually model parameters in some sense.
Yeah, and the reason, so going back to your observation that actually this type of modeling is frustrating because you have to assume lots of things have certain values and those are hard to measure. The thing we're assuming here is that what defines a cell type is the degree to which different genes are switched on and switched off. And I would say as a first pass, that is exactly what we mean by a cell type. Even, across the board, across all types of tissue, what makes a cell type is the complement of genes that it tends to express. And then, of course, a lot of other things. But as a first pass, that's how people even define cell types when they measure them empirically as well. So that assumption, I feel, is fine. As to what density each cell ends up with, that's something that can vary an enormous amount. And that's something that's seen in the data and was initially fairly puzzling. So one of the other questions we wanted to address was just how you can get so much variability, several fold variation in expression levels, while not losing a coherent cell type, if you like.
Because I guess one consequence of your model is that, say, if you have a neuron type which has, twice as much of a particular potassium channel compared to a particular sodium channel, that ratio will stay fixed, even though the overall numbers of both of them can sort of vary.
Yeah,
Is that?
yeah. So the ratio stays approximately fixed,
Yeah.
and
So
then
that's sort of
that's
what
exactly the
really.
case. Yeah.
Yeah. Ah. Well, it's very elegant. I hope it's right, because this is something I understand.
I mean, there's questions of right
Yeah.
in
Absolutely,
modeling,
of
right?
course.
It's
Yeah.
very hard to say any theoretical model in biology
Mm.
that is right. They're all wrong.
Mm.
The aspects of it that it helped us with was it helped us understand what might be the primary driver of
Mm.
cellular identity, and one idea that had been out there,
Mm.
which might have some truth in it, but I believe it less now. But one idea that was out there was that neurons must possess intricate sensors of every single thing that
Mm.
they need to keep in check. Now, that's complicated, but biology is complicated, so in principle, perhaps that happens. But it's complicated in a more troubling way, theoretically, and that is that if you have multiple processes all fighting with each other to achieve a certain balance of physiological properties, there are many, many more ways in which all of those goals can conflict with each other.
Yeah.
And you can end up with all kinds of pathologies as a result. If you just have one central regulator, you lose some of the, if you like, the precision that you would with a more detailed scheme. But what you gain is a certain amount of robustness to loss of a certain regulatory pathway, and so on. Now, I think the truth probably lies in between.
Mm.
I think the truth lies between this concept of master regulators. I think there's evidence that actually whole chunks of the genome are under the control of a large master regulatory pathway, but there is more than one of these. And then within them, there's levels to the amount of control. So in that paper, we focused on regulation of ion channel genes at the level of transcription. But of course, there's all kinds of things that happen at the protein level, at the membrane level, and so on. And we've done later work just piecing together what might work there. And the short answer is, if you've got a fairly good master regulator, then on top of that, you can have enough degrees of freedom that you can have local regulators that do even more refinement and control.
But I guess, at least when I sort of, because when when I started doing neuroscience, one of the things that puzzled me a bit was that why is there so little calcium inside cell, right? I mean, why is the neuron or cells going through so much trouble to pump out all the calcium? And of course, in this context of making a contact between the electrical properties of neurons and to what goes on inside the cell, like having this very, very low concentration to begin with of calcium makes it well set up to maybe to be this regulator, right? If it was,
Absolutely, yeah.
yeah. If it
And
was like,
again,
yeah.
the cell biology itself is a constraint here because calcium is toxic
Ah,
in large quantities. So
yeah.
it's very heavily regulated in organelles inside the cell, in endoplasmic reticulum, in mitochondria, and so on. So it's extremely tightly regulated. This makes it a good signal because the signal to noise ratio is favorable. But as a result, or possibly resulting from that, there is this heavy constraint that actually it needs to be on average in a fairly tight range for the cell to even survive.
Huh. So at least sort of when this, so you show in this paper that if you had just different choices of this time constant or these rates, then you can sort of make all kinds of like type 1 and all kinds of like type 1, type 2 and bursters and pacemakers and all this rich variety. So, but one question is sort of, can these rate constants change over time? Or
Yeah. Yeah.
so
So
yeah, there's like many time scales, of course, in neuroscience. So
yeah. So to get at that question, we need to understand what these rate constants might mean. So for people listening, what we're talking about here is the rate at which something like an mRNA is produced, and then as a function of mRNA concentration, how much protein is produced. And then there are other steps. The protein's floating around, but how quickly is it inserted into the membrane and assembled into tetramers and so on? So there's a lot of levels to this, and they have characteristic timescales. And as you point out, um, we assumed, just in this very sort of spherical cow type model, that those rate constants were fixed for a cell type. But we did ask the question, what happens if you change them? Why might they change? Well, let's think what they mean. If you think about gene transcription, this could be the affinity of a transcription factor to its binding site. For example, that's something that would alter the rate. If you think about translation, it could be the strength of ribosome binding to the RNA. And again, that's something that can change with context. So all of these things can be regulated and are regulated by the cell. So there's no reason to believe that these rates are absolutely fixed in time or in space. They could change. On average, probably there's some sort of characteristic rate. But if they change, then this gives the neuron ability to kind of steer around in conductance space more effectively. But this now brings us back to that original problem of what's to stop it getting lost. And as you start tinkering with these different rates, depending on the context that the cell is in, it could find itself essentially maintaining its set point, but its properties are not nominal for the circuit that it's in. For example, it may be aberrantly excitable. It may lose excitability, for example. So you could lose circuit function catastrophically in this way if this process isn't very carefully controlled and possibly doesn't have the aid of other sensing mechanisms as well. So we certainly don't want to and didn't want to convey that it's all just as simple as master regulator, fixed rates, and everything works fine. As a first pass, that does a good job. But there is certainly the possibility for pathologies to occur. And that was really the closing part of the paper, where we explored, well, what happens if you delete something? What are the type of recovery options available for these different cell types? And broadly speaking, the answer is sometimes they'll recover.
yeah.
Sometimes they'll partially recover. Sometimes you can inflict some kind of insult to the cell, and even if in principle it has the right remaining ion channel complement to compensate for it, because of its regulation rule, it won't find it. And then you'll get some sort of aberrant effect like hyperexcitability. And I didn't feel this was something we needed to fix in the model because it's something that happens in the biology.
Yeah.
So we know, for example, in something like chronic pain, there are insults that occur in the nervous system that result in aberrant excitability. And it would be wonderful if our nervous system and the neurons in it could steer themselves out of that, but they don't. Evidently, they can't. And it's likely that there's, the underlying regulatory pathways are, largely intact. Maybe there's something missing from them. It's likely that the neurons are capable of expressing the right kinds of proteins to steer themselves out of this mess, but they don't, because essentially they can't see where they are. That's my interpretation of this through this very simple model.
Because also, I think, isn't it in like, if you look at development of healthy development, then you also have like neurons in the beginning. Don't they have a lot of GABA, or there's some like different systematic differences between ion channel densities, right? So there's also a change of like these
Yeah,
ion channel densities as you develop. So maybe
absolutely.
this also reflects some changes in this, in some time constants in there or whatever in the.
Yeah. Yeah, absolutely. And we made a very toy model of that in the paper. And essentially, we grew a CPG network, and
CPG. What is that?
so a
What?
central pattern generator network, which is really the basis of the neural nervous system that Eve Marder studies in her
Yeah.
lab. She
So
studies
that's like this.
these motor circuits, which their sole job really is to generate a very stable motor pattern and modulate it in an appropriate way in different
That's
contexts.
in the crab, right? Or in the lobster or crab, right?
Both the crab and the lobster, so the
Okay.
crustacean
Yeah.
stomatogastric ganglion.
Okay.
So in the paper, just as a sort of proof of principle, we asked, could we take 3 motor units that are initially not excitable? They aren't at their calcium target, and we just set them off from random initial conditions where there's not many channels expressed in the membrane. A little bit like you would imagine a neural progenitor cell. They're not particularly excitable, but there are channels present in the membrane. And then they have this program in them that says, right, we're going to insert channels at these various different rates until we get to a set point. It turns out that if you, without actually much tweaking, you can make a self-organizing rule where each of these cells just monitors its own calcium, and then the network converges on a really stable triphasic rhythm. So
Okay.
you can program essentially a motor pattern in the genome. That's the high-level interpretation of this, which is ultimately how this must work, right? The stomatogastric ganglion doesn't learn by trial and error how to move the muscles around, because the crab would be long dead if it had to do that. It knows instinctively how to generate this motor pattern, if you like. And one thing we found was, yes, you can do this, but then once you've got this mature network, if you perturb it, it doesn't always reject that perturbation in an ideal way. It doesn't necessarily preserve its oscillation frequency, for example. But if you now switch the rule, so if you like, the neurons started off in their immature state, they get to the network and then something clicks, and now they have a new set of rates, then they'll do a better job of maintaining that motor pattern.
Wow.
So you're absolutely right. These, at least observationally, the complement of excitable properties of a cell certainly changes over development. It's entirely plausible that there is one type of logic regulating it when it's a small nascent neuron, and that logic switches as it reaches maturity. I don't actually know much about that. This again seemed like a reasonable cartoon picture of things.
So when you think of this, like the intracellular calcium, then as a regulator, I mean, I guess it could be both be. I mean that transcription happens inside, at least in mammals, happens inside the nucleus or inside
Yes.
the yeah. So it could be like the calcium concentration inside the nucleus, or it could be intracellular calcium outside the nucleus, right? Or in other parts, right? So I guess can a model say anything about
No,
this?
so
Or?
so the model is fairly agnostic about that. I mean, actually, the way I think about it is that there is if you like, a kind of latent state in the model which represents the general activity of some transcriptional network. This is the thing we're calling the master regulator. So it's not calcium concentration per se, but it's something that is directly coupled to calcium concentration that's doing the job here. So that's the element, if you like, that has the memory of what's happened over the last minutes to hours of the cell, and is sort of tracking whether or not it's on target or not. And that could be that could be implemented in any number of biochemical entities, but they would have to have there would have to be essentially a large enough copy number of them to store this signal faithfully. Nice thing about calcium is, even though it's there at low concentrations, the number of calcium ions inside a neuron is an astronomical number. That's not true for things like even RNAs of certain kinds. The count could be quite low. So if you actually want to build a good homeostatic regulation mechanism, the best way to do it is to use essentially an integrator, so a cellular memory that has as its main storage molecule something that's present in high numbers inside the cell. And small molecules are good for that. Large molecules like RNA is not necessarily that good for that purpose.
Like to fluctuating or whatever.
Indeed, yes.
Yeah. So then if you look at down at the genetic level, it must be, so I guess like these neurons, that it must be some kind of a regulating part of the DNA which is then different or which are differently. Yes, how okay, so how are these differences encoded on the DNA level?
Now you're going to hit the boundary of my knowledge.
Okay.
So what I'm going to do is give you the version of this that I would tell someone down the pub, where they don't have a smartphone on them right at their hand to sort of fact check me.
Okay.
Broadly speaking, the genome of a cell is decorated with all kinds of additional molecules like methyl groups and so on. And in the case of like a mammalian cell, the chromatin around DNA is also modified. And what this does is it makes certain genes accessible or inaccessible. And it can really have the effect of completely shutting down a subset of genes. And that's a sort of extreme example of this. So you can have a cell which has the same genome but have very different modification of its genome that makes certain genes usable or not. And then on top of that, you can have different splice variants and so on of the genes that are actually turned into RNA. And those can be modified and acted on. So a simple example is something like RNA interference. So there are things like microRNAs inside neurons, and there's actually quite a lot of those in neurons, and those can inhibit the mRNA message, if you like, and target RNAs for being broken down and so on. So if you think about what all of these things are, all of them impinge some way on the overall rate of production of any of these signals. So a simple way of modeling this is to just say, right, some signal affects the rate of production or the rate of degradation of each of these entities. How it happens will depend on which entity that is, where it is, and the cellular context. So again, it's a slightly waffly answer. It basically says biology has a lot of tricks up its sleeve in order to do this. So I have no doubt that it does happen.
Yeah. So this is like under the umbrella of like epigenetics, isn't it?
Yeah.
So this is all
epigenetics is
like
a broad term to describe this. But even if you talk outside the epigenome, for example, you just talk about the transcriptome, that itself is regulated by many processes, and then the proteome itself is regulated. If you think about where these channels end up, they end up inside vesicles inside the cell. They're shuttled around. And whether or not they even make it to the membrane or not depends on a whole other set of factors. So there are many places where a cell, including neurons, can intervene to control these things.
Cool. So I think one of the many things that you learn from this paper, and one is that like you can have like you can measure from a neuron over a long time, and it looks like it behaves the same. a
Mm-hmm.
reliable burster, for example. But then that doesn't mean that the ion channel composition in that neuron is the same, right? So
That's
this is
right.
like
Yes.
this dynamic. So it's not really the ion channel that's conserved. It's the like the function. And I guess
Yeah.
this led you over to this idea of representational drift. So you had this paper, and the first paper I read from you, maybe it was you and Michael Rule and Christopher Harvey, I guess, in 2019.
Yeah.
So you had this nice paper where you started with, well, it's like quite literary and literary sort of references. Heraclitus of Ephesus is quoted as saying that one cannot step into the same river twice. So how is that? What is
Mm.
representational drift, and
Okay.
how does
Yeah.
this the quote relate to it?
Yeah. There's quite a leap here.
Mm.
There's a leap in themes, but there's not really. So there's a leap in sort of neuroscience themes, but not really a leap in content and concepts here. In the same way that you can have a neuron with a well-defined set of properties underpinned by vastly different combinations of effectors, or in this case, ion channels. In a neural circuit, that's even more true, if you like. There are so many more degrees of freedom in a neural circuit. If we go back to thinking about the central pattern generator, the cellular properties themselves can change. The connectivity can change fairly dramatically. And yet you can still have a fairly consistent overall function. Now, there's limits
Mm.
to that. And actually, in the case of central pattern generators, the connectivity has a very strong determining effect on the type of pattern you can have and how robust that pattern is.
Okay.
On the other hand, if you have a large neural circuit which essentially uh, stores associations by modifying the receptive fields of neurons, and it does this throughout an animal's life, and for good reason. These receptive fields need to be able to change to accommodate changes in the animal's
So
environment.
then let's just go. Yeah. So what's the receptive field, just to make sure that everyone
Yes,
is on board?
sorry. So, um, receptive field, I think, classically it applies to sensory neurons, where you would find a neuron, for example, in a visual part of the brain, and show an animal a visual stimulus, and you will ask, well, what aspect of the stimulus maximally excites this neuron? And the classic example of that comes from Hubel and Wiesel, where they show, in this case, it's an anesthetized cat, uh, oriented bars, so bright bars on a dark background. And as they change the orientation of those bars or the position of them, they find that neurons have, if you like, a combination of stimuli that maximally excites them. So if you map out how the neuron responds as a function of all these different combinations of stimuli, their orientation, their contrast, their velocity, and so on, you build up a map, and that map is the receptive field. And it's a fairly general concept because you can replace the notion of a visual stimulus with an auditory stimulus, or something completely abstract, like a conjunction of stimuli that are represented deep inside the brain, or even not a stimulus, but an output. So the same type of receptive field is found in motor units, for example, in primary motor cortex, that essentially there's a maximal direction that certain motor neurons tend to fire as an animal is moving its arm, for example. So that's the concept of receptive field. And receptive fields are built up essentially by neuron tuning the relative strengths of different synapses coming in. And by biasing away from some and towards others, it can reconfigure its map, or rather its receptive field, so that it listens more to certain subsets of inputs than others. And this by and large, is the basis of most models of learning and memory, of that modification process. And then, a static snapshot of that kind of tells us what different neurons represent either internally, externally, and then in this way, we sort of understand something about how to map function in the external world into neural firing rates and so on in the internal world in the brain. But what I've just described, this process of essentially pruning and strengthening different inputs, it's fairly intuitive that there are many ways I can do that while preserving my overall response properties. So that's one thing to consider. And then if we step back from that, and instead of thinking about the detailed response properties of individual neurons, if we just think about the overall transformation of what happens when you put the information into a neural circuit and you look at what comes out, there's no reason for those receptive fields, if we describe them, to actually remain the same anyway. You can preserve the overall transformation while allowing those receptive fields to move around somewhat. So that's the idea that motivates this concept of representational drift. But the concept didn't really arise just by thinking about it. It arose from observations of this actually happening in different parts of the nervous system. And one of the reasons Chris Harvey was involved in that review that we wrote, where we sort of christened this, and we weren't the first to point this out, by the way, that there are other papers going back more than a decade pointing out the receptive fields aren't necessarily stable. One of the reasons Chris was involved in this was that his group had identified or provided one of the starkest examples of this, where they showed. So his graduate student at the time, Laura Driscoll, had performed these really tough experiments. She'd trained mice in a virtual reality tea-maze, which by itself is actually not a straightforward task. You've got to be pretty patient, and you've got to have a lot of opportunity for a mouse to get good at this and even to play ball with you.
So what's the task?
The task is that the mouse runs in this virtual treadmill, and it's in a virtual reality environment. It sees a visual stimulus at the start of this virtual maze, and the maze is shaped like a T. So it runs down this long corridor, and at the end, there's a T junction. It can turn left or right. And to get a reward, the mouse has to turn left or right, depending on which stimulus it saw at the start. There's two stimuli. So they learn to do this, and it requires, obviously, visually guided movement and some sort of short-term memory to remember what the cue was, and correct association. So it tests a lot of skills, if you like. And Laura was studying part of the cortex called the posterior parietal cortex, which is right at the top of the mouse's brain, making it very accessible to, in this case, it was 2-photon calcium imaging. And the advantage of this is that you can see the responses of cells, and there's no doubt as to which cell really you're looking at, because the recording is very high resolution, and each recording session it's possible to register the microscope, so it's looking at the same field of view. And again, sweeping a lot of technicalities under the rug, which are non-trivial, you can track cells over many days. So that's what she did. And to their surprise, they found that animals that had learned this task and were performing it essentially at expert level, day in, day out, over the course of a month, that the response properties of these PPC neurons had two interesting features. So number one, you could pretty much reconstruct the entire trajectory of a mouse just by looking at which neurons were responding at which point in time. Almost like what people refer to as a place code or a place field code that's seen in the hippocampus, for those listeners who are familiar with that. In other words, that at each point in the track, there was a neuron or a subset of neurons that got excited and sort of said, right, we're here, we're in my favorite place. And then the rest of the track, they were more or less silent. And in this way, you could sort of map out where the animal is and what it's doing. So this seems to indicate that the PPC is doing something fairly strongly related to that track. And indeed, if you shut down the PPC, the animal's performance on this maze task collapses. But over the course of about a month, the responses reconfigure significantly, such that those neurons that were encoding certain parts of the maze on day 1 might have disappeared altogether by day 30, or they might be encoding something different. So there was this dramatic reconfiguration of the code to the point where if you mapped it on day 1 and tried to decode on day 30, you'd have a real hard time. If you wanted to actually in detail pick out where the animal is. You could decode things like what its overall decision was, but it was very difficult to decode things like the location or the speed the mouse was traveling.
Because this
So
is,
that's an example of drift, because the animal is not learning during this time, in a sense. It's not getting any better on this task.
So exactly, because, and I think a key thing here was really the technical breakthrough of being able to record this for so long time and making sure that you're actually listening to the same cells. And I mean, if you're measuring with electrodes, then you could sort of, well, maybe the electrode is moving, and then.
Indeed.
But what about? It could also be state dependent, couldn't it? I mean, that's also something to control for, or that is something different. Yeah.
Yeah, so we would argue that it is indeed state dependent, and we just don't know what that state is. And one possible state is time itself. And there have been studies since then trying to dissociate whether or not time, as in wall clock time, elapsed amount of time since day 1, how much drift that accounts for. Or whether it's experience during the task. Those two things are different. And the evidence now suggests that it's a bit of both. That both experience and elapsed time contribute to drift in slightly different ways. That evidence largely comes from other circuits, though. It comes from places like the hippocampus. So there is indeed something accounting for this reconfiguration, but it doesn't find itself among the typical factors that an experimentalist measures, such as what the animal is doing, its performance on the task, and other things like that. If you do more fine-grained measurements and look at things like individual muscle movements and so on, you can account for a chunk more of this variability in some cases as well. But it seems that there is always a chunk that's not accounted for. And indeed, this is the simplest explanation, is that there are spare degrees of freedom in the configuration space inside the brain. And over time, it starts to move into them.
I guess also, I mean, if you look at what is known about, I mean, many of these, I mean, synapses are on spines, right? And the spine dynamics, that is, I mean, the spines on the dendrites are changing on this time scale. So somehow it must update something, right?
Yeah. There's several lines of evidence and related phenomena here. So what you're mentioning now is kind of zooming down a level to the sort of microscopic processes that are underlying this. And indeed, in many parts of the brain, the dendritic spines, which are protrusions that are the sites of excitatory synapses on certain types of neurons. So they're a proxy for connections. These things naively, and in some sense the dogma used to be, that once you had a large mature spine, it formed a very strong, stable connection. That is largely true, that there is a sort of differential stability of these spines, so the big ones tend to be more stable. But there is far more turnover than you might expect in the sort of intermediate range, where there are functional connections that are just coming and going all of the time. And if you think about it, this is entirely consistent with our view of cellular turnover in basically all tissue. That cells essentially need to break themselves down and repair themselves continuously. There's this balance between anabolism and catabolism right across the board. And that's just to maintain proteostasis and other things. And really, the way a cell achieves growth in a particular direction is by biasing the balance between destruction and assembly. That's how these spines move, in fact. So actin polymerization is what drives this. And it's a balance between breaking down actin filaments and building them up that actually causes the spines to move. So if you like, it's this kind of balance between formation and degradation all the way down. And you might ask why essentially the nervous system doesn't dispense with this at some level. And it does. So there are structures that are incredibly stable, right?
Okay.
There are axons in my midbrain and my spinal cord that go right the way through my body. And they stay there most of my life. They transmit information, hopefully, reliably. In the case of primary motor afferents, they impinge on the same neuromuscular end plate. And if that's cut away, it can grow back to exactly the same spot. So when biology needs to ensure some kind of consistency, it has that capacity to do so. But for whatever reason, in parts of the brain, there's this fairly frenetic turnover in connections. And that doesn't necessarily mean that the effective connections change, because I can have 3 or 4 connections between neuron A and neuron B, and I could lose 2 of them in one place and gain 2 in another. And by and large, this likely accounts for a lot of turnover. But it still doesn't answer the questions of why there's turnover. And a simple answer is, if you want a system to be flexible and plastic, the best way to achieve rapid response is to have this ongoing push-pull and just modify it, right? Because the overall rate of change is the difference between the push and the pull. So it gives you far greater range to steer your system. So if I want to rapidly upgrade connections to a neuron or downgrade others, if I'm already in the process of building some and destroying others, then all I need to do is stop building or stop destroying, and this will have the effect of shifting the connection in the way that I choose. I being the neuron in this case. So that's my answer for why there's so much turnover, is that these circuits need to be able to reconfigure. So while they are maintaining, if you like, relatively stable overall properties,
I think we lost the
they're
connection
just
here.
keeping a balance between the breakdown and the formation of these connections. That has interesting side effects, though. And the interesting side effect is, if I keep my overall function, but I have this turnover, I will necessarily wander around in configuration space.
Yeah, I guess this idea of having, and just like with this balanced excitation inhibition idea, that you have all this ongoing excitation and inhibition also makes the neuronal responses faster than if they were silent to begin with.
Yes, that's
so I
right.
guess it's a little bit of a similar
indeed.
idea, maybe.
And actually, it's something that again, going all the way back to molecular biology and cell biology, this is something the synthetic biologists found early on, is if I want to turn genes on and turn genes off quickly, the thing I target is the degradation rate.
I see, yeah.
That's the thing that controls the time constant of the system. And if I have to have a high degradation rate, I have no choice but to have a high synthesis rate as well.
Yeah. So, okay, so one thing with this, if you have this high dimensional space of possibilities and this low dimensional, relatively speaking, behavior, then we know that it's okay to change you can sort of keep your function and change the parameter. Of
doesn't
course,
tell
that
you
doesn't,
how, though.
No, and it doesn't, and of course, No, that's true. And one option would be to say, okay, I can do it, but I'd rather stay fixed. I
Yes,
mean,
that's the safe option. Yeah,
you could do that also, but this
yeah,
is, but you have this idea that it's actually this drift is an expected consequence of maybe learning and
yeah,
like the other synaptic
yeah,
processes that
exactly.
goes on. Yeah.
So what I would say is, I don't think any of these ideas hold to the exclusion of other possibilities. I
Mm,
think some connections are likely essentially hardwired in all parts of the brain.
mm,
But we've got to account for the things that aren't, because these can and do have an effect on behavior.
mm,
Um, so what I would say is that if I had a neural circuit and I wanted to uh, remember or to store a function faithfully, I
mm,
have a couple of options. One of them is to program that function or learn it early on, and then just make sure nothing changes.
mm,
Now, that's the approach we use if we're designing an electrical circuit.
mm,
Um, but even when we design an electrical circuit, we ensure that that overall function is preserved within the tolerance of the parameters
mm.
in the circuit, such as temperature tolerance. And this is something people
Mm,
don't tend to think about enough,
mm,
is that
mm,
actually, no, that circuit isn't the same. It does change, but we
mm,
designed it in a way that it's robust to those changes. So that's one option. Another option is to um, give the circuit the tools to learn this function itself.
mm,
And for many kinds of function, that's what we think happens in the nervous system.
mm,
It's a strange idea, though, that we should have a circuit that has this capacity to learn, and then somehow it realizes it doesn't need to learn anymore, so it just stops.
mm,
There's something circular about that, right?
mm,
In a sense, the only way it can know that it should stop learning is essentially if all of its predictions always come true,
mm,
and that seems unlikely in the real
mm.
world. So
Yeah.
my guess is that a lot of neural circuits have a sort of basal rate of learning,
Mm,
and this keeps them in check. That
mm,
learning is of course shaped and sculpted in a way that it doesn't cause dramatic reconfiguration every time there's something new.
mm,
Some of that might be, for example, relegated to certain circuits like the hippocampus. Certain circuits are remarkably plastic and buffer these major changes. But it's quite possible that large parts of the brain are just always learning.
mm,
You can't stop that.
mm,
And that continual learning contributes small perturbations to the underlying configuration and the underlying structure, including
mm,
things like the receptive fields or response properties of neurons. And the drift that we see is a hallmark of that,
mm.
that it's just
Yeah.
this ongoing learning ticking away, moving the circuit through this configuration space.
So then there are like even when you don't get any kind of new sensor input, there is this
Mm.
continuous. This idea is that this continuous communication between different brain areas where they
Yes.
sort of predict and get like you because you tie this to this idea of predictive coding, like
Yeah.
internal predictive coding,
Yes.
where they sort of send this. Because I mean sometimes I just remember I saw this movie that what's called one of these. It was like an old comedy. I think it was silent movie with Mel, whatever. One of these old movies, and then it was like some of these. I remember some. I like that word. It was a funny movie, and I remember some of the scenes. And when I saw it again, I
Mm-hmm.
sort of I remember the scenes, but I mixed up the actors. So the actors were sort of like standing differently compared to what how I remembered it. And of course, that was not very important. It was equally funny,
Well, this happens to me on a daily basis,
you know,
and
yeah,
it drives my family crazy that
really,
I
yeah.
frequently I remember things that happen, but not necessarily the sequence in which they occurred,
Yeah.
which can get me in
So
trouble.
that's true.
Um, so yeah,
Yeah,
maybe this is a an artifact of that.
of this.
I don't know.
No,
That's something where I'd be really
yeah, no. I mean that's
speculating.
sort of but at least it's sort of it has the same kind of qualitative features
Yeah.
at the
Well,
behavioral level.
actually, a nice thing that it does sort of prompt is it forces you to ask, well, especially in the case of an organism like a mouse, which is the favorite organism of large parts of neurobiology, does a mouse really need to remember what happened to it 6 weeks ago in a fairly, benign environment where nothing
Yeah.
dangerous happened to it or threatening? Probably
No.
not. So it would be surprising if you had this sort of savant-like capability where a mouse can kind of remember every detail of a task.
Savant
Um,
mouse.
that seems odd. And, just anecdotally, it's not true in humans either. We're pretty lousy witnesses if we're asked to recall precisely what happened to us 2 weeks ago on a certain day. Some people can do it, but most people are not very good at that. And that's consistent with the idea that actually there's a large amount of turnover and reconfiguration of the various forms of attractors and structures that form memories. Yet, what's interesting is we don't we tend not to forget the important things. So this for me is actually one of the most interesting questions, is how you organize learning and memory in neural circuits so that they have this graceful property that the stuff that's important to you on a 2-hour time scale gets remembered for about 2 hours. And the stuff that's important for you throughout your life tends to be remembered on that time scale as well. I don't think we have good answers for that. And I think that's a really important question that lots of these observations, I think, prompt.
I mean, you had this other paper that I also read together with Miku, I think.
Yes, Charlie.
Miku,
Yes.
Charlie, and from 2023, where you went
Yes.
a little bit further into this idea of representational drift, and then you have also had this nice quote. This was actually, "I am tomorrow or some future day.
Yes. So
What I
James Joyce.
established
Yeah.
today, I am today what I established yesterday on some previous day." So,
Yeah.
I mean, you have a sort of a Irish sounding name? Do you have some James? Do you have an Irish connection or?
No, I have I do have an Irish connection. I also have an internet connection, so I can find
Okay.
nice quotes. But no, the basic idea, I think, in all of these things is that, and it is not. So Michael was instrumental actually in the first review and sourced this specific quote about stepping in the same river twice. I didn't know who said that, in fact.
So,
But
oh,
I like the general dialectic viewpoint in biology. I think it's a useful one. I think a lot of biological processes can be explained by opposing forces and shifting the balance between these opposing forces. Lots of things make sense once you adopt that viewpoint. And the other one, the James Joyce quote, basically saying that we're not the same person each day. I think is very real at a deep psychological level, and that's a useful perspective because it forces us to question this rather mechanical assumption that you get the feeling that we've had in neuroscience, especially in theoretical neuroscience, where certain things should be stable, otherwise they're no good. The real question is not how do you make something like a memory stable forever. The question is how do you make memory useful forever? And those are not the same thing. So how you actually usefully adapt your memory and usefully adapt your world model is a far more important question than how do you learn it once and for all? And now you've learned it, how do you just use it? I think that's a caricature of large chunks of theoretical neuroscience, or it's a caricature of the question of what learning and memory is useful for. I would say.
But I guess also it's an example of where sort of like our metaphors from like thinking about machines
Hmm.
and the machines we make, they want to be stable, and we don't want them
Indeed.
to, yeah,
And
and the
yeah,
computer
and in
programs we make should be stable
yes,
too, right? It's the
and in
same
that
thing. If
review, we sort of comment on, that moving away from the philosophy towards more practical things, that
mm,
actually some of our scientific practice informs how we think about these things. So even just if you think about how you design an experiment to interrogate memory or learning,
mm,
you have a phase where the subject comes in and they're naive. Then you have a learning phase where you
mm,
bombard them with some information or
that's true.
whatever. You reward them for doing the right thing. Then you have a
Mm,
testing phase. So there is this baseline learning and
mm,
afterwards paradigm throughout. Now, that's not in place because we think this is, a natural model of learning and memory. It's in place because of the scientific method.
mm,
And it's in place because of ways of thinking about repeat experiments that in the end we borrow from physics and so on, where
mm,
you establish a baseline, you do a perturbation, and you look what changed.
yeah.
That can easily leak into your thinking.
Mm,
And as a result, you can end up really thinking, well, this is how it works because this is how we study it.
mm,
The same is true on the theoretical side. If you look at even from the beginning, the earliest to the most recent models for what learning and memory are, you have your naive neural network, you have your training phase, you get to a criterion where it's learned, and now you stop the learning and
mm,
the network performs its function,
mm,
which is exactly like the experimental paradigm. So if
mm,
we continually think that way, then it's easy to fall into the trap of thinking that, well, this is how the biology works as well.
yeah.
And it doesn't.
Mm, mm. Interesting point. Also, I think in this like this 2023 paper with what's it called, Charlie Miku, you had this really cool idea. I think with where you sort of because we're used to thinking about learning like with these learning
Mm-hmm.
rules that you somehow, well, you move upwards in the fitness landscape by these
Yes.
algorithms, and then but you're saying that actually with the learning rules that are typically used, these stochastic learning
Mm-hmm.
algorithms, you, in addition to going maybe upwards and becoming a better fit, having
Yeah,
upwards in the fitness landscape, as a side effect you move also sideways
that's
in
right.
this landscape, so which
Yes,
means that it's drift, right, equally good, but it's, so
absolutely.
that's an interesting idea. So how, so what what is a stochastic learning algorithm? Maybe start
Right.
talk a little bit about that.
So, if you've got large amounts, well, first of all, let's talk about what a non-stochastic learning algorithm is. Let's say
Mm,
I've got a bunch of labeled data and I want a model to spit out my Y label when I give it an X label and so on. I take all of my
mm,
data, I compute my loss. So I look at the mean squared deviation or something like that between my model's current prediction and the actual answer. And I greedily minimize that loss, maybe by following a gradient. Now I've got a choice. I can compute that gradient across the entire data set.
mm,
And if it's a really small data set, that's fine. And then I will find a nice optimum. There might be more than one, but I will find one conditioned on the type of um, optimization that I do. But if I have a really large data set, or let's say I have a rolling data set where there's data
mm,
coming in and there's data going out, I can't do that anymore. I have
mm,
to do my learning with a subset of the data, hopefully a representative subset. So I'll take a subset of these points and I'll do the same thing, but the direction of greatest ascent or descent in an error landscape will point in a slightly oblique direction. Even if I have a really great, nice, even
mm,
sampling, I'll point down the true hill, if you like, but slightly away from the absolute steepest direction. And
mm,
then as I reach my minimum, I'll essentially reach a coastline that surrounds the level that's essentially unobtainable because I take finite steps and so
mm,
on. And as I approach that coastline, the effect of these lateral kicks will become more and more apparent because the gradient steps get smaller and smaller. And now basically all that's left are these inconsequential steps that move me along the error surface. That is drift, in my view. And what's interesting, and what we dug up in that paper, is that that exact phenomenon had been studied in mainstream machine learning and neural networks. And there were some papers that we dug up that had done that, and had been studied indeed by computational neurosciences. So Alexei Kulikov had a really nice paper pointing this out as well. For me, one of the things that's really interesting about this is I can do this kind of stochastic gradient descent in an entirely deterministic way, right?
Okay.
You can think of the phenomenon I just described as, there's just a bit of noisy perturbation going on and inching me along this neutral error landscape. And it doesn't have to be the gradient learning rule that does that, right? Any noisy perturbation, for example, a bit of turnover in the synapses, if it's a real neural circuit, will do the same thing, okay? But I could take a very large data set and just sample with replacement from it in a deterministic way. And on a shortish timescale, I'll still get drift. So it's not the stochastic bit of it per se. It's the fact that I'm not seeing the full picture.
Okay.
And it's the fact that the true gradient, if you like, is unobtainable. And I would argue that that is the case in the real world. When we're trying to learn a model of the real world, what's the chance we're going to get every detail right? It's zero. So we're always going to be aiming, hopefully, in roughly the right direction, but slightly oblique to it.
That's true.
And it's that oblique component that's going to shift us around forever on some kind of level set in error or performance.
So I guess if you sort of have like this dog detector and the only thing you see for a while are poodles, then your dog detector will be affected, right? That if you become less poodle-like, then a Siberian Husky might not be recognized.
And I'm sure that's the case. I don't know about dogs per se, but I'm definitely better at recognizing things that I'm familiar with than things from the same class that I'm less familiar with.
Cool. So so these things, I mean, is it the more you learn, the more you drift? Is that a consequence of this?
Okay. There's a couple of relationships. So the picture I've just painted, and the review will do a better job of explaining it. It's all down on the page with diagrams. Says that we essentially have a random walk going on this surface of low error. And this surface, let's be clear, it's a hypersurface in a very high dimensional space. So it's a manifold with many, many directions. So if I have a random walk on a manifold like that, I'll very quickly lose my origin, right? If I do a random walk on a line, with probability 1, I'll get back to the origin. It's also true in 2D. It's not true in 3D and upwards. So I'm just going to shoot off into the unknown, even though it's just a kind of Brownian motion. So that's the picture that we have. And time inevitably is a factor here, right? So the thing I've just described is a diffusion. So the mean squared deviation grows linearly with time. No getting around that. As long as I change nothing about the underlying process, mean squared deviation grows linearly. So as a function of time, my configuration is just going to move. So time should have a factor. Now, what else will affect this? Well, if any of the drift comes from learning itself, and by learning, we're now going to paraphrase this as sort of gradient descent or optimization, or something equivalent to that. Well, there's two main factors there that will determine the amount of drift that I get, at least in the toy model where I'm using gradient descent, and that is the learning rate, so the size of the step that I take, and what's called the batch size. So the batch size is the number of data points relative to the full data set, if you like. And fairly intuitively, if I have a really large batch of data, then my gradient is going to be really well aligned with the true gradient. So my oblique step is going to be quite small. If, on the other hand, I take just a couple of samples, then I'm going to get a gradient direction that's probably quite misaligned with the true gradient, and it's going to push me along the coastline a bit more.
I think
so. This, if you like, is like the most basic first principles model for drift, and it tells you the amount of experience should affect it. So limited experience should cause more drift. The rate of learning should affect it. Now, I don't know what controls that parameter. I suppose this could be intrinsic to the subject. Some people learn perhaps at a higher rate than others. It could be related to arousal, engagement, the amount of reward that's received, so on. And then time. Time is fairly clear. So even in that simple model, at least 3 factors affect drift. There's a fourth one too that's implicit in all this, which is how big this, how the dimension of this manifold that I've just described. As I increase the dimension, the overall distance that I move from the origin scales as well. Okay. I've got more dimensions to walk in. And in this cartoon model, the dimension of that manifold is essentially the spare degrees of freedom that I have to learn a particular task within relative to the number of degrees of freedom I have in a neural network. So the size of my brain, if you like, relative to the complexity of the task is another thing, another factor that affects the drift rate. So task complexity should scale drift rate as well. Drift should be slower in more complex tasks that have been learned properly. A complex task that's been improperly learned could result in higher drift because it's following.
Has there been any attempts to look at this experimentally?
People are trying, yeah. So I've been speaking to a number of experimental groups, and they are trying to measure these things and vary these precise quantities. So I know that Yaniv Ziv's group in Weissman is directly addressing these questions, the question of task complexity and the question of time versus experience and so on. So yeah, there's this something that people do take seriously, and there are fairly clear predictions. I think it'll be interesting to see what happens. I think we're still waiting.
So I mean, one feature that our brain luckily have when they work is that you don't have this what's called catastrophic forgetting that you have with this.
AI except networks. When we do.
Yeah, exactly. Well, that's true. That's also true. But at least it's not maybe. Well, yeah, it doesn't. The story is that it's worse in AI, right? So, is there anything that what would be your advice to AI people in sort of improving their algorithms?
Well, I mean, I don't have a lot of deep wisdom here. I mean, just from scanning the literature, lots of the ideas, the obvious ideas, are being tried and tested. So one of them is that you actually formally compute the sensitivity of, so say you're going to make a weight update in a neural network. What you want to do is figure out which weights are most likely to change your performance on the task that you've learned. And that's something you can compute. So you can compute the sensitivity. In fact, you will have computed it already if you've learned a task. And what you can then do is, if you want to learn a new task, is partition your weight changes to ensure that you only affect the directions in weight space that haven't been used much.
Okay.
So that idea has been out there for a while. It works. It can be computationally heavy because you have to compute second order properties of, so essentially Hessians or approximations to Hessians. Another idea is to actually not throw away all of the tasks that you've learned. So if you want to learn 3 tasks, learn one, learn another, but then switch back to the other one, and so on, and keep learning.
Yeah.
So there's a number of ideas that are already out there, and they essentially all amount to kind of factoring and compressing, if you like, the information that you've learned in a way that new information is either incorporated in a consistent way or it's kind of given its own place. And at a high level, that makes complete sense. The details of how you do it efficiently, I think that's still an active area of research. But for sure, studying this in animals can provide clues. I would be very wary of assuming that the animals do this perfectly, though, including humans.
Yeah. No,
What's interesting, again, is that not that we don't forget, but it's that we forget or we compensate for forgetting in a way that is compatible with our goals in life, if you like, that we forget all the time, but somehow we either reinstate those memories, reconstruct them, or preserve them in a way that tends to minimally impact our day-to-day life, and until we have a disease of memory, or we have a condition from birth that has aberrant memory phenotypes associated with it. So the biology is pretty rich. I don't think it contains the perfect solution, but I think it
one
provides
way is
really interesting clues.
Like old things are, maybe there are parts. I mean, as you mentioned, there are like, I think also you say in a paper that like songbirds, they don't have a high flexibility in their brain. They don't drift a lot.
Right.
And I guess
Well, this
also
is what we're told. Yeah. So from speaking to people who study songbird learning, they say that the song is learned in adolescence and crystallized, and the response properties of the neurons in the various parts of the brain are super stable, and that's manifested with a certain amount of rigidity in the animal's behavior later in life. So maybe that makes sense.
But maybe also, since if you sort of look at people with dementia, for example, they typically they have a harder time actually remembering the recent things than the real old things. And so maybe the old things, like first day at school or whatever, is actually stored in a way with less drift or something. So
so again, this is wandering off topic where I have any expertise, but my informal thoughts on this are there's two. So one is a disease like Alzheimer's disproportionately affects the limbic system. So places like the hippocampus and so on, they start going downhill sooner than others, and we know that recent episodic memory is heavily dependent on the hippocampus, whereas procedural memory and things like that is less hippocampal dependent. So that can explain part of it. But beyond that, I think you're right that as we learn, we use formative experiences as a scaffold for later experience. So we form a kind of personal world model that is intrinsically linked with early memories in childhood and so on. So maybe it is that actually more of our sort of procedural memories, a bit of an episodic component gets woven up into them or something like that. I don't know, but I think that there probably are good explanations for this.
I guess in principle, it could be tested in mice, where like certain memories are more important than others, and like the memory of food or enemies or
absolutely. And those are the kinds of things I'd really like to see, and I'd really like to see this kind of longitudinal study of not just the behavioral response, but the neural response as well. And I think we've got to be very careful about that as well. Like detecting the memory is often shorthand for they had a certain freezing threshold in a task or something like that. That doesn't mean that an animal remembered something or not in the way that we think. Similarly, a neural circuit response, just because you reinstate a certain pattern of activity, doesn't mean that the animal can access it consciously necessarily. So I think all of this has to be studied in lots of different dimensions. But adding this component of what happens over extended time periods is something that I think we should push towards. Of course, it's very challenging. It's not straightforward to do this.
So we're getting closer to the end here, but in some of like communication before this podcast, you said that you were a bit that this representational drift term was sort of it that you somehow, I don't know, regretted it that you sort of coined it in this paper because it had been sort of misused or misunderstood or something.
Well, yeah. Plenty of people have said they don't like the term for various reasons.
Okay.
Some people don't like the word representation.
Okay, yeah, no, that's
it's a reserved word, right? It has a highly specific use, for example, in certain parts of psychology. Actually, in mathematics, it has an extremely specific use, and it's very clear we don't mean that. And other people argue that actually just seeing a neural response is not the same as seeing a neural representation. I agree with that. I think that's true. So that's a problematic word. And then drift is a problematic word because for some people, that means, oh, it's just random. Actually, I think it's a bit more clear cut than that, because the reason I like the word drift and proposed it is it's an analogy with the drift diffusion equation, that there's a systematic component to that, which is the drift bit, and the diffusion bit is the non-systematic part. But, this isn't going to convince people who just don't like the word.
It's a representational diffusion. That's
you could call it that. I don't know. But it's two words. They're fairly catchy. I think that annoys people too. I think the other thing that happens is, as with lots of words, people bring their own interpretation to them. And this happens a lot in neuroscience, actually. We have this sort of morass of terminology where lots of it is extremely context specific, yet we abuse the context specific component of it all the time, and we throw these words around fairly casually. So that's an aspect to this. I think any name we would have come up with would have annoyed somebody. So I'm not too bothered about that. I'd like to get past it though, and just say, okay, it's a thing. It's not a perfect name, but we'd like to give it a well-defined meaning, just so that we all know what we're talking about, and that I think is far more important.
Than the word itself. Yeah, cool. So now, anything on your mind? Anything we didn't discuss, which you would like to? This is your opportunity.
No, I mean, I've enjoyed this chat, and I think what I appreciate is actually teasing out a fairly obscure journey from studying single ion channels all the way up to asking whether or not we're the same person today as we were yesterday. I hope that hasn't been too confusing for anyone who decides to listen to this. But yeah, I just appreciate all the questions you've asked. And one thing I would say is that, and this is personal to everything we've talked about, that in neuroscience, and especially in theoretical neuroscience, there's this odd division or dividing line between people studying what they would call biophysics. Sometimes this gets the moniker detailed modeling, which I think is actually problematic, versus people who study other things. I think this is unhelpful. I think you can see the same theoretical concepts reemerge over and over and over again at all parts of the nervous system. I don't think that there is a preordained best level to study things at. I think it's good to study at all levels. And one of the real antidotes for this, at least in my career so far, has been to teach and interact in other disciplines, including engineering, but also in cell biology. So I teach a course where I teach actually some of the biology of E. coli.
Okay.
And one of the remarkable things about this organism is, under the right conditions, it's capable of sensing, moving. It has a primitive form of memory. It can make decisions. It can do many of the things that we attribute to nervous systems. And if you ask what the mechanism is, or what the circuit is that underlies this, the answer is it's a bunch of biochemical reactions. And for me, this really illustrates the point that if you want to study autonomous behavior, let's say, and this is an example of autonomous behavior, or sensing and acting, and so on, you can study it at that scale and still have something that resembles a form of cognition, if you like. Now, a lot of people are going to, spit out their tea if I use that word in the same sentence as something like E. coli. But if you actually study how these organisms interact with their environment, at a high level, they perform some of the functions that animals with large nervous systems perform, and they do it in much the same way. So I think we can tease out many of these principles, not independent of the level, but actually ascending and descending levels, and actually being a bit more level agnostic and inviting in these examples instead of drawing fences around research and saying, well, I don't care about that bit. I only care about this. This goes way back to the point at the start of the discussion about interdisciplinary science.
Yeah. Well, I certainly agree with you. It's not, and at the least sort of detailed model is better than a realistic model. That's really bad, because that who knows what's realistic. So because lots of detailed models are not realistic. So it could have been worse.
We agree on that, and yes, we also agree there's never a perfect word for anything.
No, so but I certainly at this stage of the field, right, with neuroscience, and it's like it's really unclear where the most successful breakthroughs will be, and it's like what level it will be and how it will. So I think we just have to be open to all kinds of approaches at different levels and so on. So that's at least sort of that's why I even had a podcast here with what's called is the guy in Marcus Covert, who has like made a model for a bacteria.
Oh yes. No, I've chatted. I know Marcus. I've chatted to him. That was a pretty ambitious.
That was a very interesting thing, and sort of
enterprise.
So I'm certainly very I mean, inclusive kind of approach to this podcast. At least I try to.
I think that's the way. Yeah.
Yeah. Very good. Well, thanks a lot for the conversation, Tim. So it was really a pleasure, and good luck to you and the future of representational drift and all the new things that
we'll drift onto something else pretty soon, I think.
or maybe diffuse into something else.
Thank you.
Good.
Okay. Bye, Gota.